think we're about 30 participants already, so that's great. Um, so I'd like to welcome everybody to the Roy Behnke Department of Internal Medicine Grand Rounds. We appreciate everyone's attendance today. We have a real treat today with Dr. Scott Burgin from our very own USF Department of Neurology. Dr. Burgin is a professor of neurology, and he is in the cerebral vascular division of neurology, where he participates on the stroke service and has been the fellowship director for several tours of the cerebral vascular fellowship in the Department of Neurology. Dr. Burgin hailed from the North Carolina area, with graduating from Duke University, a Blue Devil in his undergraduate days. Uh, then headed over to UNC Chapel Hill, where he completed his medical school. Then off to Rochester for his neurology training, which then concluded with an NIH-funded cerebrovascular fellowship at UT Houston. He made his way back to Rochester, his beloved Rochester, and we were able to have Scott come down to Tampa in 2011, and he's been here for the last dozen years, being part of a very, very vibrant Department of Neurology. His CV includes numerous clinical trials, publications, abstract poster presentations, textbook chapters. We are truly blessed to have Dr. Burgin give us what is our first neurology grand rounds of the year. So we've been itching for one, and we're getting one. So Dr. Burgin is going to speak on molecular biomarkers for ischemic stroke. And we welcome you, Dr. Burgin, with open arms as a Department of Internal Medicine. So looking for you to talk. Go for it.
Well, thank you, Dr. Sennett, and everyone for the invitation. It's been a couple years since I spoke at Medicine Grand Rounds. It's going to have to say pre-COVID, because yes, I had to trek up north and got lost finding the conference room. But I'm really looking forward to this opportunity to speak. I'm going to talk about something that's really become incredibly interesting to me over my career. I actually started right out of fellowship trying to look for biomarkers for ischemic stroke. And I guess I was a little bit before my time, but I didn't ever really lose my interest in this area of biomarkers. It's not just about stroke, but it's about everything that has to do with what they are and what they mean and how do we find them. And so I just wanted a chance to go over some of the work that we've been doing at USF and Tampa General. A couple little teasers for things to come, because as Lance Armstrong said, it's not about the bike. This really isn't necessarily about stroke, but it's about sort of the voyage we take in medicine and trying to do a better job at what we do. So if I can figure out how to make my slides move forward. It really comes down to why I'm particularly interested in a stroke biomarker, is that we really need fast and accurate ways to diagnose a stroke. This may seem a little bit basic, but just to give people a background, stroke is a really very significant medical problem. It's the number 2 cause of death worldwide. Fortunately, we've gone down to number 5 in the United States, but still it's the number 2 cause of death in the world, and remains everywhere, US, international, the number 1 cause of disability. And in stroke, we're kind of in an interesting situation where we have a lot of things that look the same on the outside, but look very different on the inside. And so when it comes to stroke, we can generally break things into 2 categories, kind of I have in this little cartoon over to the right. About 15% of strokes are caused by bleeding into the brain, around the brain, the hemorrhagic strokes, with the remaining 85% being ischemic strokes, which is from a reduction or complete cessation of blood flow. And as a current counting, I think there are at least over 300 causes that can contribute to ischemic stroke. And so this presents quite a diagnostic challenge. A lot of these patients look exactly the same on the outside. We will all know from any clinical time that there's many mimics of things that can look like a cerebral infarct. It can be seizures, it can be migraine, it can be tumors, it can be alcohol and drug intoxication, it can be metabolic, it could be, you name it. I guess if there were 300 causes of ischemic stroke, there's probably 3,000 causes of things that that can mimic a stroke. And we've not really gotten much better over the years at diagnosing this. And the reason we need to diagnose this is that rapid treatments in the area of particularly ischemic stroke, what I'm mostly going to talk about today, are really very, very critical time-based diagnoses. Right now, we're pretty much still limited to our clinical exam, which is worth a lot. I don't want to downplay that. And a couple of very high-tech brain scans, CT scans, CT angiograms, CT perfusion scans, high-tech MRI technology. But the reality is, for most people in the US and even more so in the world, and this is a worldwide problem, there's really a general limited expertise out there in a timely manner to be making these diagnoses. And there's a technical expertise. You can't have a fancy functional MRI machine out in the middle of, rural Montana. It's just not really practical. And as again, if we look at ourselves as world citizens, this is a big diagnostic challenge. So what we really would benefit from is something that is an accurate biomarker that's really practically usable and available to a wide population. So this comes down to a question really then to begin with, what is a biomarker? And it's really just a fancy way of saying it's some kind of biological characteristic, biomarker, that we can observe outside the patient. And this isn't a new thing. I mean, this is really part and parcel of a lot of what we do. These can be molecular, histologic, radiologic, physiologic, ranging everything from your blood glucose to a lung biopsy to even your blood pressure. These are biomarkers that are readily available. And we've learned over the years that it's really important to be able to make these measurable characteristics because people are different. And we really need ways to tease apart what's going on and what's happening in order to provide care that's really personalized to our patients. But one thing, and I'm going to bring this up more than once because I think it's important and it's easy to kind of go down the biomarker rabbit hole, is that it's really important that personalized medicine is personal. Don't forget that in the end of the day, what we're treating are patients and people, not their biomarkers. Although you might kind of catch me at times saying that we need to focus on that, but it usually needs to be taken with the caveat that you always got to track this back to the patient comes first. So there are a number of available biomarkers categories out there. This is a picture in the top right from the FDA. They have a branch that's particularly focused on biomarkers. And we can generally classify them into 7 categories that you see depicted up here. And for stroke as an example, you can translate this to almost anything you want, but as stroke, we have a need in all 7 of these categories. For example, there's susceptibility and risk biomarkers. Could we tell who's going to have a stroke or when are they going to have a stroke? Would we be able to take some kind of test to be able to determine if a patient's going to respond to acute therapy like thrombolytics or preventative therapies? How are they going to respond to their statins? In most of what I'm going to focus today is on a diagnostic marker. Do we have a test that can tell a stroke from all these 3,000 other things that can look like a stroke? All of the things that come in and cause a stroke alert in the emergency department. Can we have a simple test that divides those into stroke versus not stroke? Could we have a test that tells us something about the cause of a stroke? Is it a large vessel occlusion? Is it a lacunar stroke? Can it tell us anything about when the stroke happened, a stroke clock? A lot of our treatments are very time sensitive, and we base them upon when was the last time we knew someone was doing well That's a really terrible criteria for trying to treat someone with a life-threatening illness, and you can't do anything because you don't know when it happened. And could we have markers that give us information about people's risk factors? You know, do we have a blood test for atrial fibrillation? Could we have a blood test that tells us something about how hypertension has contributed to the cause of their event? It'd be nice to have markers that let us get a better judge of safety. Could we have tests that tell people who are going to have complications? Could we tell the person who's going to have a bleeding transformation from thrombolytic drugs? Do we have a blood test or other test that would tell us if someone's going to develop serious brain swelling and brain herniation down the road? Could we get some tests that help us monitor patients better? You know, right now we go and do neuro checks and people's vital signs, but, you know, could we get a test that tells us how someone is responding to reperfusion therapy? Did they open their artery up? Is someone going to be able to have a test that tells us that someone's about to go down the tubes? Is brain edema about to kick in? Are they going into vasospasm after subarachnoid hemorrhage? From a kind of a practical clinical trial science basis, we would like markers that can tell us about responses in pharmacodynamics, because really developing this marker gives us really an insight into the mechanism behind disease. Would we be able to better plan clinical trials? Would we better be able to plan interventions? And if we're good enough at this, we can even get biomarkers that act as surrogate endpoints, which will allow us to develop quicker, faster, and cheaper clinical trials by being able to find an endpoint, a biomarker endpoint, that is early, that reliably predicts events down the road, so we don't have to do such long and involved clinical trials. Just an example of this has been a sea change in things like multiple sclerosis, where in the early immunomodulatory trials, we had to follow people for years to see if these drugs had an impact on their long-term outcome. But they did a lot of work and found that plaque burden measured on MRI is an excellent surrogate endpoint for long-term disability. So there's been a plethora of new drugs and trials that came out where we were able to do this quicker and faster. All we had to do was show we could get the plaque burden down, and then people do better. And now we have 30 drugs on the market where we used to only have 2. And then lastly, we would like something that would be helpful for prognosis. Could we tell how severe a stroke is? Could we give information to the family about how well we think someone's going to do, not just, gray hair Dr. Burgin going in there and stroking his chin and saying, I think you're going to do poorly. What we've learned, those are not very accurate ways to assess people's prognosis. And could we come up with a test that predicts people's chance for having a recurrent event? And that's it. You can take this and put whatever part of medicine you're interested in, and you can see there's a need in all of these 7 categories. But when you're developing a biomarker, you really need to think a bit about what the advantages and disadvantages are of a biomarker, just knowing what you're dealing with. The good things about a biomarker is it's usually a measurable entity. We can look at it with some objectivity. We can look at it with an eye upon the precision of the test. We can do things to determine how reliable and how valid it is. And as said, it can give us a view of mechanistic insight that we didn't otherwise have. But developing these things is not so easy. We've learned over the years that, much like anything, timing is critical. You do a test at the wrong time, it might not tell you what you want. You wait too late, it might not tell you what you want. Developing these things and implementing these things are expensive in both time and resources. Just like everything, they're subject to sample storage and lab errors. You let it sit out on the counter and it doesn't work. You take it to the lab. There's a chance that it's not going to work. You can drop the tube and your blood goes all over the floor. Not perfect, but once we have these, it still can be difficult to establish the range. And no matter what anybody says, anything we do has some sampling risks. Checking your pulse, very, very low risk, but a biomarker like a brain biopsy carry some very obvious sampling risks. So we have to consider all of these things when we're trying to develop one of these new instruments. So in looking at a career of trying to develop a molecular stroke biomarker, I kind of came up with a wish list. What would I like to have if I could look in, sort of have my magic crystal ball and be able to do this? And in trying to develop this, we wanted to find something that was universally available, something that's available in the hospital, but something that could be done in the field, in ambulances, in urgent care centers, in various healthcare places all over the world. We wanted something that was minimally invasive and low risk, of course. And for this, blood is really quite the optimal source there, and what we're mostly focusing on here. As we learned very early in medicine, really usually the challenge for patients is keeping blood inside the patient, not getting it outside of the patient. So it represents a really good, readily available place to look. We wanted something that was simple, portable, and durable. It's not really good to have a test system that if you knock it off the counter, it's irreparably broken. We need something that's hardy. I really would love to have something that gives information about a temporal profile to be able to help make treatment decisions. I wanted something with rapid and comprehensible results. It's not really good to have a test result that you have to have a PhD to make heads or tails out of what it means. Optimally, you want the kind of thing where you pee on a stick, and if it's got a plus sign, it means you've got condition X, and if it's got a minus sign, it means you don't. We wanted something that allowed for sequential serial assessments, so we can get a time course, something that maybe we don't do just once, but we're able to gather more information by doing sequential testing. And of course, we want something that doesn't cost a lot. So looking at my wish list, some of those things are easier than others. So it comes down to the question, how do you take that wish list and turn it into a practical tool? And this is just a cartoon I got from actually a renal journal that I thought did a good job of explaining it. And so really we start with discovery. We look at basic science. We look at tissue markers that we may have found that look interesting in bodily fluids or tissues. And we kind of have to use our knowledge and our basis to sort of prioritize and think of those that we think are more important than others. Then we have to take that idea and develop an assay. And in doing that, we have to develop an assay. We have to test this assay against existing samples and establish disease to really determine the test characteristics, the sensitivity, the specificity, the ROC curve, the positive, negative predictive value, and such. And a lot of ideas, great ideas, make it to this point, but unfortunately don't make it to this next yellow box, which is clinical validation. And this is where the rubber meets the road. So we have this great idea. Well, we really need to take this and put it in a real world population and see if this biomarker accomplishes one of the 7 goals that we set out to establish. And this is the tough part. But if we get through this part, we really do have a tool that we can use and apply to help take care of our patients. And really, this is the step towards personalized medicine. But again, I just want to caution again, remember, what we're doing here is really, we always need to take this back to the patient. And really remember, we're treating the patient, not the patient's numbers. But those patient's numbers really can mean something and allow us to do some meaningful things. So let's take this back to stroke. So in the very beginning, well, if we're looking for a biomarker for stroke, where do we look? And the answer is basic science. And I'm going to get on my soapbox here and say, when people begin to question what's the value of doing basic science, even if it seems to be unrelated to anything immediately at hand, is that basic science is the reason we're here doing what we're doing, and it's the reason that we're going to have a career going forward, and it's the reason that we will continue to make discoveries. This is really important stuff. And so in this case, we really kind of looked at some basic science. And this little cartoon on the left is not on the test and is by no means comprehensive, but just to give an example of some interesting molecules and entities that have been discovered over the years. Some of these may be more or less familiar to people, but in the case of the nervous system, there are a lot of interesting entities in the oligodendrocytes and the neurons and astrocytes and the parasites in the actual endothelium and in the blood. And so all of these are interesting targets in places that people have looked. Each one of these, is several, PhD people's, dissertations in entire careers here. This stuff means something. It's just a matter of how do we use it. But in stroke, it gets a little bit more complicated because not only are we dealing with all of these pathways and things going on in stroke, it's very time sensitive. We have hyperacute, acute, subacute, and chronic stages of stroke where some of these pathways are important and some of these pathways are less important, things that turn on and things that turn off. And, so we have to not only consider the marker, but where in the process of the injury that we're dealing with our patients. So it makes this a little bit more of a complicated moving target. So this is really where the quest for the stroke marker Holy Grail begins, is looking at these markers. And this is just some data from a really quite good review article from a few years back that reviewed 150 studies that were trying to do this very thing. And where people did was we started with simple molecular markers. All of those little letter number combinations that you saw in the previous cartoon, which can be peptides and cytokines and stem cell markers and other things, is a good place to look. And we would just start with picking one. And I just picked S100 GFAP out of the list because glial fibrillary acidic protein seemed like a great place to start. Just on the surface, it's a cytoskeletal element. It's almost exclusively in CNS astrocytes. You injure the brain, it's only really in the brain. If it goes up, maybe you have a stroke. If it doesn't, maybe you don't. Well, it made sense to a lot of people, but unfortunately it didn't work out quite like that. In a meta-analysis, just picking this GFAP example, looking at 11 studies of almost 1,300 participants, well, GFAP levels, which are relatively easy to measure, could distinguish a stroke from a hemorrhage. Not something we have a hard time doing with a CAT scan, but it could do that at less than 3 hours, but not so much at less than 1 hour. It really couldn't tell. It also couldn't tell a stroke from something that looked like a stroke because GFAP levels also go up in tumors, traumatic brain injury, seizures, etc. So it may be too simple. It's not like the little Dutch boy being able to put his finger in the hole in the dike to keep all of the sea from pouring into the Netherlands. Single markers may lack the sensitivity and specificity to really be able to accurately reflect the huge diversity of things that can appear like a stroke. So what did people start to do? Well, let's start mixing and matching these markers. Maybe if we come up with a combination of a few of these, we can assess a couple of different pathways. Maybe we can catch people at different times, and maybe we can have improved test characteristics for a test. Great idea. Been successful in other areas, not so much in stroke. There's been really no clear winners to date in this approach. There was a study a few years back in 2017. I love the name Stroke Chip. I thought this was going to be it. They did absolutely everything correctly. They went into the literature and found the 21 highest likely culprits to be a stroke biomarker. They put them in this relatively easy to deal with chip. They did a multi-center, prospective, international, collaborative trial of 1,300 participants. They were enrolling people under 6 hours, trying to decipher a stroke versus a mimic. Also, they looked at stroke versus hemorrhage. They took these 21, they did 500 subjects, went back and picked the handful that they thought made the most sense, boiled those down, and did a validation in almost 800 subjects. And these are the results. And I just want to draw your attention down here to the bottom right corner. I'm going to be talking a lot about AUCs, areas under the curve, receiver operator curves. This is not the biostatistician's favorite test because it makes a lot of assumptions and is not perfect. But really, it's a good way when we're talking about a bunch of these studies to come up with a rough way to try to give them some sort of equivalence for comparison. And with these ROC curves, there's usually a number, and 0.5 would be the equivalent of flipping a coin. 0.5 to 0.7 is generally considered a discrimination. 0.7 to 0.8, acceptable. 0.8 to 0.9 is generally considered excellent, and greater than 0.9 is outstanding. And so for this trial, their cutoff for success was to come up with AUC that was 0.8 or greater. And this is what they got with their best panel. They found for the best panel, comparing stroke versus mimics, the AUC was 0.74. For stroke versus intracerebral hemorrhage, 0.76. Not meeting their criteria. And actually looking, the panel didn't do much better than some of their optimal individual markers that they looked at. So this was rather disappointing results for this protein panel biomarker approach. So people got to thinking, maybe proteins aren't the way we need to be looking. Maybe RNA would be what we need to look at. And the thing about it is that RNA changes occur for a lot of types of injury to the brain, and almost every step from the brain all the way through into the blood. All of these ischemic changes have essentially been associated with changes in RNA expression, which may be detectable in the blood. And a lot of these markers that are happening in the brain do find a way to make it out into the blood. So this is a new approach. This seems like a really great way to go. There's some challenges in testing blood for RNA. Number one, it's kind of messy, as everyone is aware. It requires some rather nuanced analysis. The blood carries a lot of immune system cells that are spitting out stuff. And really, if you think about it, the blood practically carries everything else in the body as well. There's a lot going on there. There is the fact that the brain is protected from the circulation by the blood-brain barrier. So some brain markers may only make it out into the systemic circulation at low levels and have very faint signals, or some may not make it. But it is rather easily obtainable and testable. And a lot of these stroke mechanisms, we know from the basic sciences that they do result in at least some degree of identifiable and measurable changes in the peripheral blood. So it seemed like a good place to look. It's got other advantages and disadvantages as well. Back in medical school, there was kind of like we talked about coding MRA and junk MRA. I think we were a little over enthusiastic in calling it junk here. There's the coding RNA, the messenger RNA, which is the ones that lead to protein. That's the one that I'm going to be mostly focusing on. But there's a lot of other interesting non-coding RNA. We've got long non-coding RNA. We've got these small non-coding RNA, like the microRNA, which are fascinating. We're looking at expanding a bit more into this, but I think this is the next thing to come. And the good thing about, though, these RNAs is that they're rapidly expressed. We see RNA changes well in advance of any protein synthesis because that's what drives it. And secondly, some of these RNAs, including the microRNAs, non-coding RNAs, they function as signaling molecules and protein modifiers. So they can be subtle bits of information that we could use for diagnostic and clinical purposes. But there are problems. I mean, it's really historically been very difficult to deal with this. We typically have had to know what the gene was we were interested in to go looking for the RNA. Detecting fold changes of less than 2 were historically very difficult. There's a large amount of noise in the blood, especially if we're getting any kind of cell lysis from collecting the blood, dumping all kinds of intracellular things out into our tube, and such that these loud, systemic RNAs could mask the quiet, subtle ones we're looking for. And from a practical perspective, this is rather a slow and expensive process to examine RNA, but seemed a very promising next step. And so what people did was really take this approach and run with it. And they developed these oligonucleotide microarrays in ways to try to test these in larger populations. But when we looked at what had happened with strokes, all of these studies had limitations. In most cases, the microRNA, the oligonucleotide microarrays, again, they derived them from genes that they already knew about. At most, technically, they could look at up to 40 maybe genes at any given time. Most of the studies that were done were comparing strokes versus controls, i.e. healthy people walking around out in the community who volunteered. Most of these were trying to distinguish a stroke from a hemorrhage, something where it's important, but not really what we were looking at. And many of these trials were really very small, averaging somewhere between 15 and 40 subjects in a study. A lot of them didn't look early on in stroke. They were looking at stroke at 24 hours and comparing ischemic strokes versus community controls. Not really the target we were looking for, or really what we really had a practical need for. And what I've included here is just an example from a good paper that was well done study. They took 18 genes, compared ischemic strokes to, again, 20 community controls. And what they found was that in about 66% of the time, it could distinguish a stroke from a control in 66% of patients in under 3 hours. Not so great. At 24 hours, it could tell the difference, but in a practical perspective, that really is not a very useful test. There's been a larger one. This array technology has gotten a little bit better. There was an Affymetrix microarray study done, again, very well, multi-center, only looked really got people within 24 hours, but they got 1,700 subjects, 66 controls. I just want to say it took them about 3 or 4 years to collect these 1,700 subjects. But the analysis, again, was not very strong results. It could, excluding lacunar strokes, cryptogenic strokes, TIAs, and mimics, it could tell stroke versus controls with an AUC under 86. And notice there was kind of a caveat in what it was able to do. Sensitivity, okay. Specificity, only about 0.6. So still room for improvement, not quite ready for prime time. So this was all history getting us to where our journey really began here at our sleepy little university by the sea. So what we did was we looked at all this and said, look, we need a research plan if we're going to find a messenger RNA stroke biomarker that is able to accomplish what these really excellent scientists did ahead of us. And to do this, we had to come up with several sort of baseline assumptions that we had to work with. Number one, we considered that strokes, the transcripts from these could come from practically anywhere. We weren't going to be really exclusive about that. We were going to use cutting edge analytic and computational resources, next generation RNA sequencing, quantitative real-time PCR, This would allow us to detect fold changes much lower than historical approaches, and we could probably come up with results with higher accuracy. We also thought it was really important to look at early suspected strokes. I don't have a hard time telling someone who had a stroke 2 days after they had it. What about people when they hit the door? And can we detail the stroke, not from someone in the community, but someone who looks like they're having a stroke? And we wanted bigger sample sizes, larger sample sizes than the previous studies, because as the statisticians all know, more is always better. And so our primary aim, it's good to have a primary aim, was to see if we could come up with an acute stroke diagnostic blood test that would reliably distinguish stroke mimics versus strokes. But our secondary aim, we weren't on blindsided, was maybe we might discover some interesting things along the way. So we broke it down into 2 steps. The first step, very much from the beginning, in blood, we wanted to identify RNAs that differ between ischemic strokes and mimics. So in effect, when we take someone who has this, clinical signs of a stroke, have this RNA that predicts this picture on the right, which is a large right middle cerebral artery infarct on CT scan. Can we do that? But once we did that, we actually have to make something practical. Can we develop an automated system that can measure these identified markers in all the ways that we said we wanted from before? So this is how we did it. And there's, again, no test on this. And I'm going to state this as I am not a molecular biologist. I'm just a molecular biology fan. But this is how, in a nutshell, we distinguished differential gene expression patterns between 2 populations using messenger RNA. So what we did, we began when people hit the door, under IRB approval, we would collect blood in an RNA stabilization tube, one that would let this kind of fickle RNA be preserved. It can keep it at room temperature for like 5 days. I can keep it in the fridge for 2 weeks. I can freeze it. Depending on how I freeze it, it'll stay good for up to 5 years. Collected these in the tubes, and stuck them in the freezer, completely blind to what those blood results were, and then collected a series of clinical and demographic data throughout the hospital course, looking at all the things we were interested in, risk factors, et cetera, and finally the adjudicated diagnosis of, was this a stroke, or was this a stroke mimic, what was the type of stroke, what was the type of mimic, et cetera. But how we actually do the molecular part of this is fascinating. I'm sure there's 5 Nobel Prizes sitting here on this cartoon. But basically what we do is we take the blood in this tube, take it back to the lab, and basically extract the total RNA. And in our case, we were interested in messenger RNA, which you may remember has this kind of convenient poly A tail. So we can come up with the complementary sequence to all these A's, and we can extract the messenger RNA from this mash of otherwise other non-coding RNA. We can isolate that. And then what we do in the first step is actually convert that messenger RNA to DNA, because it's a little bit sturdier thing to deal with. It's easier to measure. And if we know the DNA sequence, we know what the RNA sequence is. Take this otherwise kind of long RNA, of which there's 20, 30,000, I don't know how many of these things floating around in your bloodstream at any given time, but they're too long to analyze, so we chop them into a bunch of little random snippets that the machine can read, 100 to 200 base pairs. We attach these amazing little gadgets at the end called adapters, which can be barcoded, they're tagged. And what it allows us to do is one of the most amazing things out there is something called sequencing by synthesis. And so we have a $1 million science machine effectively, which takes all of these little snippets, plates them out on a plate, and then through amazing powers of computing and lasers and technology is able to do automated sequencing by synthesis, which allows the computer to read the base pair sequence of all of these hundreds of thousands of little snippets that are floating around in the blood. And what the computer does is spits out basically a little text file that gives us what the code is for each and every one of those little snippets it sequenced. And what we can do is there's a lot of quality control buried all throughout this. I'm making it seem over simple. But what we can do is take a series of software and go back to the human genome and figure out what genes these snippets are associated with. And by comparing two populations, we can look at differentially expressed genes. You know, between two samples, in this one, gene A is up and gene B is down, and gene C maybe is about the same between the two groups. And it gives us all of these, but we get them for the tens of thousands of genes that the machine was able to sequence. But what we can then do is do all kinds of interesting analyses to go and try to find out what the real differences are between the two populations. And I'm going to talk about that again in a minute. So this is what we did. This was our phase 1 study we performed in the Tampa General Emergency Department. We enrolled people with suspected stroke who we knew within 12 hours when their time of onset was. And we excluded hemorrhages, and there's a reason for that, which we'll get to. But in the end, in the first stage of this, we collected 74 ischemic strokes and 68 mimics. And for comparison, 21 healthy controls. And if you look to see that these populations were reasonably well matched, the mimics were a bit younger. That's not a big surprise. The mimics were a little bit heavier, and the strokes had a higher rate of atrial fibrillation, which also doesn't come as a big surprise, but we're able to control for all these variables. And so what we do is we take each one of these genes, gene A, gene B, gene C, gene Z, and then we can basically do statistical analysis comparing between, my computer keeps going nuts on its own, comparison. And we use 3 statistical techniques. And what we were able to do is say statistically between these 3 groups, between these 2 groups, stroke and mimic, we identified somewhere between 150 and 250 differentially expressed genes between the 2 populations. And we use 3 techniques, and as you can see, there's a fairly large overlap between the 3. And when we started to look at what do these genes do, we found that many of these genes were involved in the immune response, but a lot of them were also associated with brain and endothelial function, and some of them were doing things we really don't know what that gene was. We found novel genes in the process. And this is just an example of 2 of the genes that we had. There's nothing particularly special. I just kind of picked one. Gene 1 is actually a novel gene, one we weren't really expecting to find. And as you can see here, gene 1, which I call Willis, named after Circular Willis, because I like the name Willis. I'm calling him Willis. That isn't really his name. But you can see here that there was a gene that had a higher expression in stroke patients. But let's take gene 2. I didn't name gene 2. If anyone has a good name, please put it in the chat. But for gene 2, we actually found a marker that was increased in the mimics, not in the strokes. Interesting. So we can do this process with all these 250 genes that we had. It does get down to be important that we need to look at these subgroups that we had, because there is a lot of diversity. As you can see on the left, these are all the different types of mimics that we saw. And these are some of the big stroke categories we saw. So we have a wide variety. And this heterogeneity, just for the reasons we talked about before, can make finding an individual specific marker difficult because it may be in one stroke stum type and not another. And this is just an example to show you, again, just this is another random gene. And you can just see over here in the sort of the right column that patients with large vessel occlusion versus patients who had non-large vessel occlusion had differential gene expression in this one particular gene. So it's a nuanced analysis. And this gets screen is just to say why it was really important for us to compare the strokes to the mimics, something a lot of these trials didn't do, who were comparing to healthy controls. Because we found many genes that distinguish strokes versus controls didn't distinguish a stroke versus a mimic. Here's 3 again, somewhat arbitrary examples that I had from that cartoon: S100A, MMP9, ARG1. And you can see in all these cases, like, oh my goodness, this probably distinguishes a difference between a stroke and a control. Well, when we threw the mimic in there, we found that all three of these initially attractive appearing markers did not reasonably distinguish between a stroke and a mimic. So, going forward, it's really important that we have to consider that these mimics are different than the controls, and I don't think that's comparing the controls is a good way to be doing these things any further. So what we did was we kind of did that mix and match thing, like we did with the protein markers. And what we did was we basically took all of these genes and defined a threshold that defined every stroke with as a stroke versus a mimic. And just as example, again, a reminder of what the ROC curve looks like. And we were able to mix and match these using sort of a artificial intelligence logic. There's a kind of an algorithm to go about this. We were able to panel these genes to come up with combinations that seemed to fit what we wanted. And depending on the stroke subtype, one panel may more accurately determine a stroke versus a mimic versus another. And this is just some examples here at the bottom, but I want to draw your attention to the NIH stroke scale score at the bottom left here. This is something that's probably our most used stroke biomarker, the National Institutes of Health stroke scale. So we said, okay, let's look at that as a biomarker. How accurate was it at detecting a stroke? Its AUC was 0.6. Really, that fell in the poor discrimination category. But let's just pick a single gene. We picked a single gene. This is an example one, AUC of 0.7. But look at the panel on the right. This was our top panel. Our AUC for this panel was greater than 0.9. Mixing and matching these genes. So maybe we are catching different pathways. Maybe we are catching these different points in time by using the right secret sauce formula of genes. And here's just, our main results from this, kind of our top diagnostic gene panels. You see all of them, these are just the top 5. Some of these have fewer and some of them have more genes. But as you can see, our accuracy is over 90%, and our AUC in some of these is 0.92, sort of creatively rounding 0.93. Seem to be very, very accurate with good degrees of sensitivity, a really good sensitivity at detecting large vessel occlusion, which we're particularly interested in because that's the target for mechanical embolectomy. So it does seem that, at least in our initial work, that blood micro mRNA panels can distinguish an ischemic stroke from a mimic in our cohort with a high accuracy. But let's take something else, right? I went on and on about a clock. How about, could this work like a clock? It seems that may be able to tell a stroke from a mimic. How about as a clock? And really imprecise timing is a big problem for us in determining treatment, and really having a clock that tells us that someone had a stroke, in a short while ago may, versus a long time ago, may really help us aid treatment decisions, coming up with something better than the last time we saw people. So we did a sub-study. This is a pre-planned sub-study, but what we did was we took this acute ischemic stroke cases who we really, really did know what time their stroke started. Either it was witnessed or someone had found them within an hour of having last been known normal. So patients within an hour of accuracy of when they had their onset of symptoms. We did linear regression and these other mathematical things that we talked about before, really to come up with panels that would be able to distinguish cases with shorter, less than 2.5 hours from time of onset, from longer than 2.5 hours from onset. Why did I pick 2.5 hours? Well, I kind of thought, well, if I'm not really sure within an hour because we gave it an hour fudge, let's pick a number that if I was even off by an hour, we would still have people within a treatment window. And we also built the classifiers really to be careful not to put anybody in the short stroke subgroup compared to the long one, because we didn't want we would prefer to make an error, not putting someone where it might be dangerous into the wrong category. And this is what we did. We found 56 patients who we actually knew what time their stroke was within 60 minutes. There were 18 shorts and 18 longs and 38 shorts. And we found that there were 210 genes that seemed interesting. And we again selected classifiers up to 12 genes. You can see that the samples were very well matched in this case. And we went through the same process. And down here, this looks familiar. This is one gene versus a panel of genes. We see that our ROC curves improve when we combine these. And our top classifier patterns showed both high sensitivity and high specificity to be able to tell an acute stroke from one that was subacute. We may be on to having a stroke clock. This is got limitations. These were really focused only on acute ischemic stroke patients. We didn't include people who came in with hemorrhages and other things. It was a smaller sample, so our confidence intervals are less. But we're pretty excited about this finding as well. So we may have an RNA panel. We found in blood markers that may distinguish between strokes and mimics and may have a clock. But we need a system. I mean, what we're talking about is doing something in a $1 million science machine in the lab. We have to have a test that's fast and accurate. And the way we did this for this study is quite laborious and slow. So what we needed was, a rapid stroke test. And qPCR is the answer. And so what we see here on the bottom right corner is an example of a microfluidic lab on a chip, not exactly the one that was developed here at USF, but a depiction of that is able to do the RNA extraction. And we were able to create primers. This is the same technology you use for your COVID test. Is we found that using quantitative PCR let us rapidly replicate the same results. This is a correlation between our sequencing results and the qPCR results with a near linear relationship. The difference is RNA sequencing takes 7 days. This took less than 45 minutes, and probably with some additional tweaking, this testing can be done in under 20 minutes. So it seems that we can take our panels that we've discovered with full genome sequencing to a qPCR technology that is both rapid and accurate. So this could easily be moved into a point of care machine. So that's where we stand. It does look like that RNA sequencing is a powerful method that we may be able to find these RNA markers that are interesting to us. And these blood panels may be able to distinguish an ischemic stroke from a mimic with high accuracy, and we may be onto a stroke clock. We need to validate the accuracy of this. We need to validate other test characteristics. And we've actually drawn a new validation cohort, which are still in the freezer, waiting for processing for us to validate both of these studies against. We're also looking into the biological molecular pathways that are involved, looking at what do these gene functions involved with, precision, repeatability, and other things. We're also in the process of developing a larger, more diverse multi-center trial. The solution to avoiding inadvertent bias is take a bigger sample in a bigger population. We're also, this next stage, we're looking at all stroke codes who come into the ED, so not excluding hemorrhages. So we're going to get more people with, urinary tract infections and other things, but that's really the robustness that we need in a test. We're looking at further subgroup analysis. Maybe this test can predict treatment response. I can't say right now, but you might want to stay tuned. Hint, hint, hint. But we're looking at stroke subtype and timing and volume and etiology. We're also very interested in looking at the scans on these people. We collected the MRI scans on all these people where we could and banked those. We've constructed a new imaging analysis system, which you see an example of below. We're waiting for the time and resources to process them through our proprietary MR system. And we're looking at all these other biomarker categories too. These are all important. And the last thing I want to say is that if you haven't figured it out, this methodology is translatable. You know, I picked stroke versus mimic. We said stroke early versus stroke late, but you could fill in the blank. You know, person comes in with confusing situation A, and we would like a tool to help us figure out if they have diagnosis B versus diagnosis C through Z. You could see we could apply this same molecular pathway to resolve those questions. So that's my presentation. I think I squeaked in right in under an hour. I have so many people to thank. This is the kind of work that it takes a village, and it involves things that I've been doing back since the early 2000s. On the left are my very valued USF and Tampa General Stroke team colleagues. I really couldn't have done this without the help of Jason Wilson and the research enrollment team in the ED. I got a lot of help from USF, Tampa General Imaging Services, the USF Supercomputer Center up on main campus. I would be remiss if I didn't talk about our fantastic corporate partner that we're in a collaborative research arrangement with, Vuessence, led by Dr. Maha Salam. I would really like to thank the Florida High Tech Corridor, who has generously supported us over the years. And I can't get by without thanking my family who've put up with me in this for oh so many years. I owe it all to them. So that's it for me. I would love any questions. If I confused anybody, I apologize, but I'm here to try to make it right.
The mics are now open if you'd like to take the mic and ask questions.
I couldn't have been that good.
Got an awesome presentation, Dr. Burgin, in the chat.
Well, thank you. Thank you. This is really exciting technology. And I'd like to say give a shout out to Tampa. This is homegrown technology. You know, Dr. Salam from Vuessence works out of USF Connect. She's in a collaborative relationship. She's USF through and through, undergrad, master's degree, PhD. And she's been working with us for about 4 years. We've gotten the great help from the university and the state. This is a really innovative approach, and I think you could pretty much see that the sky is the limit at how we could transform this. You know, ideally, I want a test that you could have at the urgent care or in an ambulance, that when they roll in the door, the EMS knew to bring the patient to the center that they needed to go to because they had a reasonably good idea what the diagnosis was, sight unseen. And in the case of the brain, time is absolutely critical. And, you know, I don't see any time in the near future we're going to be sticking MRI machines in the back of ambulances. Not that would even probably be a practical application.
Daniel Espinoza asked, is the test approved already to use at TGH?
No, this is all investigational at this point. You know, this was done under IRB approval as a clinical trial. Our first trial has closed. We're in the process of reformulating for our multi-center trial, as well as working collaboratively with a number of people through the university, including in your department, to look at ways to expand this diagnostic approach into other areas of medicine. But if you have a good idea, please drop me an email, because you'd be probably realize that there is probably something that would apply to your area of interest if we gave it just a little bit of thought.
One more question here. It says fascinating work. This is Michael Lozano. Potentially, could you look at overlapping windows? Comes in at 2 hours after LKN, will then respond better to endovascular than TPA. I hope I said that right.
Yeah, that was a little bit of my teaser on the stay tuned. Maybe we can tell people who are going to respond better with certain types of therapies than others. We're not quite ready to answer that question quite yet. How is that? But you could see that, we did not just collect one blood draw. We actually collect sequential blood draws. So we do have the capacity to be able to look at temporal profiles on these markers. But for the sake of this study, we were looking at something that we would be able to grab right when people hit the door, the equivalent of a troponin, right at the very beginning to let us have an idea if someone is having a stroke. But we have been looking at, is there a difference between time point 1, time point 2, time point 3? And part of the reason we wanted a test that was repeatable, not expensive, that we would have things that would be able to provide people temporal profiles.
Okay, that looks like it's it in chat. I often get questions by email after your talk, so I'll forward them on to you.
Excellent. Oh, wait, I got a question. I did see the chat here. Do different patient characteristics affect how they express? Yes. And what we're trying to do is that's why we collected a lot of demographic data that we're able to control for those things. And what we're looking for are markers that really are insensitive to this variability, that would allow this to be an applicable test. But there's a lot of room for nuance because sometimes these different characteristics are exactly what we want to be able to look for. Looking at people who, you could imagine a case, well, could you look at a marker that tells if someone's having their first stroke or a second stroke? Is this a stroke that is the result of someone's long-standing hypertension, or is it the result of stroke because they have new onset atrial fibrillation? All of this nuance is out there. And the good thing about collecting these large data sets, these data sets are huge, is it does allow us to put the powers of the supercomputer to work, to be able to try to sort out this nuance. But yes, it's both, something that we have to deal with and actually a source of interesting research findings.